There's a weight in the abdomen sometimes, not pain, not discomfort exactly, just a quality of fullness that has nothing to do with food, a kind of low interior gravity that pulls attention downward before the mind knows what to do with it. You've probably felt this. Most people have. They call it a gut feeling, and then they move on. Because the phrase has been used so many times it stopped meaning anything specific, but something specific is happening. Welcome to the Sleepy Biologist. And if you're here, if you found this channel and came back to it, that matters more than I know how to say simply. This space keeps existing because of the people who support it quietly. The members, the super thanks, the people who hit like not as a reflex, but because something landed. I notice all of it. And for those of you who haven't subscribed yet, there's something grounding about returning to the same place each night. That's all I'll say about that. Tonight, we're starting at the beginning of something that doesn't have an obvious beginning. A conversation that never announces itself. A process so embedded in the body's normal operations that most of human history passed without anyone suspecting it was happening at all. The gut is long. Longer than it seems possible to fit inside a human torso, somewhere between 7 and 9 meters when fully extended, coiled and folded into a space that somehow accommodates it. And along that entire length, attached to the mucosal lining, suspended in fluid, clustered in communities of extraordinary density, there are microorganisms, not invaders, not passengers exactly, residents. The number is difficult to hold in the mind without it becoming abstract. Somewhere in the range of 38 trillion microbial cells in the adult human gut, which is, it's close to the total number of human cells in the body. The two populations are roughly matched in scale, which was only confirmed with any precision in 2016 after decades of estimates that placed microbial cells at a 10-to-1 ratio over human cells. That older number turned out to be wrong. The new number is stranger in a different way. You're not mostly microbial, but you are nearly half. And that's before considering what the microbes are doing. There's something that gets quietly passed over in most biology education. The idea that the gut is hormonally active, not just digesting, not just absorbing. The gut lining produces more than 20 distinct signaling molecules. Serotonin, ghrelin, cholecystokinin, glucagon-like peptide 1. These are molecules the brain uses, the immune system uses, the cardiovascular system uses. They regulate satiety and anxiety and inflammation and memory consolidation during sleep. The gut doesn't just receive signals from the brain, it sends them. And it was sending them long before microbiology had the tools to notice what was happening at the chemical level. Long before anyone thought to ask whether something other than human cells might be involved in producing or intercepting those signals. Here is where it gets strange, and I want to stay with the strangeness for a moment rather than explain it away. Bacteria do not have brains. They don't have nervous systems, don't have anything resembling intention, don't process information in ways that map onto cognition in any meaningful sense. A single bacterial cell is, structurally, a membrane with contents, genetic material, ribosomes, enzymes, a few thousand proteins doing their specific jobs. It is extraordinarily simple compared to a human cell, and yet, the surface of that bacterial cell is covered in receptor proteins, proteins that recognize specific molecular shapes and change their own shape in response. When the right molecule arrives, when the molecular geometry fits, the receptor shifts. That shift triggers a cascade inside the cell. Gene expression changes, behavior changes. The cell does something different because something arrived at its surface. This is not thinking, but it is responding. And the distinction starts to feel thinner than it first appeared when you learn what some of those bacteria are responding to. Human cells communicate using a class of molecules called catecholamines: epinephrine, norepinephrine, dopamine. These are the molecules the body releases when it needs to shift states, when stress arrives, when attention sharpens, when the heart rate climbs, when the body prepares for something that requires its full resources. Norepinephrine in particular does not stay neatly contained within neural synapses. It diffuses. It appears in the gut lumen. the hollow interior of the intestine, in concentrations that vary with the body's stress state. Escherichia coli has receptors that respond to norepinephrine. That sentence is worth sitting with. Not a dramatic sentence. It doesn't require dramatic delivery. It's just a fact, sitting there, doing something to the shape of what you thought the gut was. The bacterium has proteins on its surface that recognize and respond to one of the primary stress-signaling molecules in the human body. When norepinephrine concentrations rise in the gut lumen, when the body is under strain, when cortisol is climbing, when sleep has been insufficient, E. coli detects that. Its gene expression shifts. In some studies it grows faster. Its virulence factors, the proteins associated with its capacity to cause harm, increase in expression. The body says, "I am under pressure," and something in the gut hears it. It's easy to feel slightly unsettled by this. That's a reasonable response. There's an intimacy to it that nothing prepared you for, the idea that the organisms sharing your body are reading your stress chemistry in real time and adjusting accordingly. But I tend to think the unsettlement comes from framing. We encounter this idea with the assumption that the bacteria are doing something to us, that they're acting on information they shouldn't have access to. It might be more accurate, or at least more biologically honest, to say that they evolved in this environment. The gut has always contained norepinephrine. The chemical landscape of the human gut has always reflected the host's internal state. Bacteria that could read that landscape, that could use it as information about what conditions were changing, had an advantage. They could prepare, adjust population behavior, survive better. This is not manipulation, it's just old, very old. Older than the category of manipulation makes sense to apply. The vagus nerve runs from the brainstem through the chest and into the abdomen. It carries signals in both directions. Roughly 80% of the fibers carry information upward, from body to brain rather than downward. The gut wall is lined with enteric neurons, a nervous system so extensive it's sometimes called the second brain, though that phrase flattens what it actually is. It's not a second brain. It's a nervous system that develops separately, embryologically, that speaks a partially shared chemical language with the central nervous system and communicates through it constantly. The gut sends signals upward through the vagus. The brain receives them and adjusts mood, appetite, immune tone, alertness. What the gut sends is partly determined by what the microbiome is producing, which is partly determined by what the microbiome is detecting, which, in some cases, is the body's own chemical state. The circle doesn't close neatly. It doesn't need to. It just keeps moving, this low interior exchange that has no beginning point and no end point, running beneath everything you think and feel and decide right now, including the quiet act of listening to this. Somewhere in your gut, a population of bacteria is doing what it has always done in the dark. Not sleeping, not waiting, just present. In the way that living things are present, reading the chemistry of the space they occupy, responding in the only way their biology allows. They don't know you're tired, but they know something has changed. There is a letter that was never meant for you, and yet you open it. Not out of malice, not even out of curiosity, simply because it arrived and your hands were already there, and the chemistry of opening was already in motion before the question of intention could arise. This is approximately what happens in the gut. Approximately, not exactly. The metaphor will only hold for a moment before the biology outpaces it. And when that happens, I'll let it go. Human cells have been producing chemical signals for somewhere in the range of 500 million years. The signaling molecules themselves, the hormones, the neuropeptides, the neurotransmitters, did not appear suddenly. They accreted slowly over geological time, each one representing a solution to a problem the body encountered and needed to manage internally. How to tell one tissue that another is under strain. How to coordinate a response across distance. How to say in molecular terms that something has changed and that change requires attention. The language is old, older than vertebrates, older than most of the phyla you'd recognize. Some of the core signaling molecules in the human body have structural cousins in organisms that diverged from our lineage. So long ago, the relationship is almost philosophical, and bacteria have been living in proximity to those molecules for just as long. Proximity is underrated as a biological force. We tend to think of evolution in terms of competition, pressure, survival. Large-scale forces acting on populations across generations, and that's accurate as far as it goes. But proximity is quieter than that. Proximity means that two things exist in the same chemical space for long enough that one begins to be shaped by the presence of the other, not through any exchange of intention, but simply because chemistry does not recognize the boundaries we draw around organisms. A molecule released by a human epithelial cell does not stop at the cell membrane and wait for permission. It diffuses into the intercellular fluid, into the gut lumen, into the layered environment that the microbiome occupies. And if a bacterial receptor protein happens to have the right geometry, if the binding site fits the way a specific shape fits a specific space, the signal lands. Not received in any conscious sense, not interpreted, just chemically acknowledged, the way a lock acknowledges a key without understanding anything about doors. What stands out to me, and has for a long time, is that this wasn't engineered. There was no moment of design. The bacterial receptors that respond to human signaling molecules weren't built to do that job. They evolved for other purposes, detecting nutrients, sensing population density, monitoring environmental conditions, and at some point in some ancestral gut, one of those receptor proteins happened to fit a human signaling molecule closely enough that the interaction was productive. The bacterium that carried it survived slightly differently, reproduced, passed the receptor on. Over time, the fit became better. The response became more refined. The signal became part of the bacterium's usable environment, a piece of information it could act on without any of this being chosen or intended or even, in any meaningful sense, noticed. It's not entirely clear where this process ends. That's the part I find genuinely unresolved, not distressing, just open, because the same logic applies in the other direction. The human gut has been colonized by microorganisms for as long as human guts have existed. The chemical environment the gut cells developed in has always included microbial byproducts, human receptor proteins, human signaling cascades, human immune responses, all of them developed in an environment already shaped by microbial chemistry. The influence runs in both directions, has always run in both directions. Serotonin is a useful example because most people have encountered the word, usually in the context of mood or antidepressants, usually in a way that makes it sound like a brain molecule that sometimes behaves badly. But serotonin is synthesized primarily in the gut. Approximately 90% of the body's total serotonin is produced by the enterochrome cells lining the intestinal wall. Not neurons, not brain tissue, but gut epithelial cells responding to chemical signals in the gut environment. Among those chemical signals, short-chain fatty acids produced by gut bacteria during fermentation. The bacteria ferment dietary fiber, and the byproducts of that fermentation stimulate enterochrome cells to produce serotonin. The bacteria are not trying to influence serotonin levels, they are digesting. The serotonin is, in a strict sense, a consequence of their presence and activity, not a goal. But the serotonin then acts on enteric neurons, affects gut motility, reaches the vagus nerve, travels upward into brainstem signaling. And something that began as microbial digestion has now touched the nervous system in ways that are still being mapped. The letter metaphor dissolves here where it should, because letters imply a sender, a recipient, a message with a specific meaning. What's happening is less legible than that, more like diffusion. The way warmth moves through a room, changing the room not because it intends to, but because that is the nature of warmth in a room. No address required, no delivery, just the slow, even spread of something that was already there. Outside, if there's any wind at all tonight, you probably can't hear it. The room is its own small climate, and inside you, right now, something ferments, something produces, serotonin probably, short-chain fatty acids certainly. Your gut lumen is busy with its ordinary work, producing molecules your nervous system will encounter before you're aware of them. The cells that line your gut were replaced, most of them within the last week. The bacteria reading their signals are the inheritors of strains that have occupied human guts for thousands of years. Both populations are temporary in their different ways. The conversation between them is not. It continues without your awareness, and without your permission, and without needing either. There was never a moment it began. There will be no moment it ends. It simply proceeds in the dark, in the warmth, in the quiet chemistry of a body that learned long ago to share its language with things that were never meant to speak it. And somehow they do. Think about what a receptor actually is, not the word, not the concept in a textbook diagram with arrows and labels, just the physical thing, a protein, a chain of amino acids folded into a specific three-dimensional shape embedded in a membrane with a region of its surface configured to recognize one particular molecular geometry above all others. That's it. That's the whole object. It doesn't think, doesn't wait. It just holds its shape in the membrane until the right molecule arrives. And when it does, when the fit occurs, the protein changes, bends slightly, shifts. And that shift is enough to start something on the other side of the membrane. There's something quietly strange about this that I keep returning to. The receptor has no awareness of what it's doing. The molecule arriving has no awareness of arriving. The interaction is just geometry. Two shapes in the same space at the same moment, compatible with each other in a way that produces a consequence. And yet this is how almost everything in the body is decided. Not by thought, not by intention, by fit. A bacterial cell membrane is not so different from a human cell membrane in its basic architecture. Lipid bilayer, embedded proteins, a boundary between inside and outside. The proteins vary, that's where the biology of different organisms diverges, but the structural logic is shared across nearly all living things, which tells you something about how old that logic is. On the surface of certain gut bacteria, there are receptor proteins. Two-component systems, mostly a sensor protein in the membrane that detects something in the external environment, and a response regulator inside the cell that changes gene expression when the sensor is activated. These systems evolved to detect things like nutrient availability, osmotic pressure, pH, population density, the chemical texture of whatever environment the bacterium currently occupies. The gut is an environment. The gut contains human signaling molecules. Some of those molecules, norepinephrine for one, happen to fit certain bacterial sensor proteins with enough precision that the sensor activates. Nobody designed this. That there was no crossover event, no moment of biological negotiation. The shape fit, that's the whole story. Adrenergic receptors in humans are proteins that recognize epinephrine and norepinephrine. They're on heart cells, lung cells, blood, vessel walls, immune cells. When the body shifts into a high alert state, these receptors activate and the tissues they sit in respond. The bacterial proteins that respond to norepinephrine are not adrenergic receptors. They don't share the same structure, don't belong to the same protein family, didn't descend from the same ancestral gene. They just also recognize norepinephrine, arrived at the same sensitivity through an entirely different evolutionary path. Two separate solutions to the same chemical presence, which happens in biology more than the tidy story of common descent sometimes suggests. Right now, the cells in your gut lining are producing small amounts of norepinephrine as part of their normal signaling activity. Nothing dramatic. Just the ordinary chemistry of a tissue with its own local nervous system doing what it does. And at the surfaces of bacterial cells nearby, sensor proteins are in their resting configuration. Waiting, in the only sense that has no awareness in it, holding their shape. If the concentration shifts, If something in your body's state changes and norepinephrine levels in the gut lumen rise, some of those sensors will activate. Gene expression will change. Populations will behave differently. Not because anything decided to respond, because the shape fit. And fitting was enough. There's something almost restful about this once the initial strangeness settles. Nothing in the body requires intention to function. Not the receptors, not the bacteria, not the signals moving between them. Everything is just chemistry finding its way in the dark without a map. The way your breathing finds its rhythm right now without you directing it. Something adjusting, something fitting, quietly in the space below thought. You know the feeling before you know the cause. The chest tightens first, usually. Or the stomach does. A quick involuntary contraction that has nothing to do with hunger and everything to do with something the nervous system registered before the thinking mind caught up. The heart rate shifts. Breathing shallows, almost imperceptibly. A faint electrical quality settles across the skin. Not quite a chill, not quite warmth, something without a clean name that the body produces in the space between recognizing a threat and deciding what to do about it. This is epinephrine arriving, norepinephrine following close behind, cortisol beginning its slower climb, mobilizing glucose, suppressing immune activity, redirecting resources towards the muscles and the heart and the brain's most alert functions. The body has been doing this for a very long time. The stress response, in its basic architecture, predates mammals. Predates vertebrates in some of its core chemical logic. The glucocorticoid receptor that cortisol binds to has structural relatives in species that diverged from the human lineage hundreds of millions of years ago. The catecholamine cascade, the adrenal surge, the autonomic shift from rest to readiness, these are old solutions, older than anxiety as a concept, older than anything resembling the situations that trigger them in a modern human lying awake at night. What most accounts of the stress response leave out, or mention briefly and then move past, is that the chemistry doesn't stay contained. It never did. There was never a version of the stress response that produced its signals only for the cells that were meant to receive them. Epinephrine enters the bloodstream. Cortisol diffuses widely. Norepinephrine, released by enteric neurons in the gut wall, appears in the gut lumen, the hollow interior space of the intestine, in concentrations that fluctuate with the body's autonomic state. The gut lumen is not a sterile corridor. It is densely populated. The microbiome lives there, in the mucus layer, in the fluid, against the epithelial surface. And it occupies the same chemical space that the stress response fills when the body shifts into high alert. So the question becomes not whether the microbiome is exposed to stress hormones during a stress response. It clearly is. The question is whether any of that exposure produces a biological consequence. And the answer, it turns out, is yes, in ways that are still being traced. The research that first made this undeniable came partly from work on norepinephrine and E. coli, which we touched on earlier, but the findings extended outward from there into something broader and harder to reduce to a single mechanism. When norepinephrine is added to bacterial cultures in concentrations similar to what appears in the gut during stress, certain gram-negative bacteria, including several species that normally maintain a kind of functional quietness in the gut, begin to express genes associated with growth and motility, and, in some strains, virulence. The bacteria are not frightened. They don't perceive the norepinephrine as a warning. They read it as a signal that conditions have changed, and they respond to changed conditions the way bacteria always respond, by adjusting gene expression to suit the new environment. If norepinephrine is present, the host is under strain. If the host is under strain, the gut environment is shifting. Motility may be changing. Immune surveillance may be altering. The mucus layer may be thinning in places. Bacterial populations that can detect this shift early have, over evolutionary time, been shaped to take advantage of it. Not maliciously, just opportunistically in the biological sense of that word, which carries no moral weight. There's something I find difficult to fully articulate about this. Not distressing exactly, more like a quiet recalibration of what the body is. Because we carry, most of us, an implicit model of the stress response as something happening to us, inside us, owned by us in some exclusive sense. The cortisol is ours, the epinephrine is ours, the chest tightness, the altered breathing, the strange electrical quality of skin under adrenaline. These feel like interior experiences, belonging entirely to the human organism, and they do belong to it. But they also leak. They diffuse into an environment that other organisms have been reading and responding to for longer than the human nervous system has existed in its current form. The stress response has always been a shared chemical event. It just wasn't legible as one until recently. The effect isn't uniform across the microbiome. This is where the biology becomes genuinely complex in a way that resists easy summarization without losing what's actually interesting about it. Different bacterial species respond differently to catecholamine exposure. Some grow faster, some slow their replication and enter a kind of holding pattern, a low activity state that allows survival under changed conditions without committing to growth. Some species appear largely unresponsive. Their sensor proteins either absent or incompatible with the molecular geometry of human stress hormones. Some, notably certain lactobacillus strains, show responses that seem to work in the opposite direction from what you might expect, not proliferating under stress conditions, but maintaining stability in ways that have measurable effects on the gut environment. So the microbiome does not respond to stress as a unit. It responds as a community of distinct populations, each with its own chemical sensitivities, each shifting in its own direction, the aggregate effect of which alters the overall composition of who is present and in what proportion. The body says something chemically, and the community rearranges itself in response. Not all at once, not uniformly, but the rearrangement is real, measurable in stool samples, detectable in the ratio of species present after a period of sustained stress versus after a period of rest. Sleep is part of this, not peripheral to it and central. The cortisol rhythm in a healthy circadian pattern shows its lowest point in the early hours of sleep and its peak in the morning just before waking. During the night, as cortisol stays low and the autonomic nervous system shifts toward parasympathetic dominance, toward rest, toward repair, the chemical environment of the gut changes accordingly. Norepinephrine in the gut lumen drops. The signal that triggered increased bacterial activity during waking hours quiets. Bacterial populations that were in states of heightened gene expression begin to settle. Some of the shifts induced by daytime stress slowly reverse. Not completely, not instantly, but the direction changes. The microbiome has its own version of what nighttime is for, not rest exactly. Bacteria don't sleep, but a different metabolic tempo, different gene expression patterns, different population behaviors. The gut at 3 in the morning is chemically different from the gut at 3 in the afternoon, and part of what drives that difference is the same cortisol rhythm that governs when you feel alert and when you feel drawn towards sleep. There is a hum in the quiet rooms that you notice only once it stops. Not the sound of anything specific, just the aggregate noise of a building settling, air moving through vents, the faint electrical presence of things plugged into walls. You don't hear it while it's happening. You hear the absence of it, briefly, when something shifts. The gut is like this, or close to it. The bacterial activity, the chemical signaling, the slow turnover of populations responding to the body's state. None of it is audible. None of it surfaces into sensation under normal conditions. It's happening below the threshold of anything you could call awareness, in the warm interior dark of a body already beginning to slow. The adrenaline from whatever today held, whatever tightened the chest or sharpened the breath, has mostly cleared by now. Cortisol is low or lowering. The signal that sent certain bacterial populations into their daytime configuration is quieting. Something in the gut is receiving that quiet the way it received the earlier noise, reading it, adjusting to it. the settling into whatever configuration nighttime allows, not with ears, not with anything resembling awareness, just chemistry, reading chemistry in the dark, in the warmth, doing what it has always done in the hours when the body finally stops asking it to keep up. You don't feel the microbiome shift. That's worth saying plainly before anything else. Whatever happens in the gut during sleep deprivation, whatever populations adjust, whatever gene expression changes, whatever chemical signals change their concentration in the lumen, none of it surfaces as a discrete sensation. You feel addicted, you feel the cortisol, you feel the particular thickness of a mind that hasn't had enough rest. The gut changes are happening underneath all of that, without announcing themselves, without any sensation you could point to and say, there, that's it, that's the microbiome responding. This is actually how most of the biology in this series works. But it feels worth naming again here, because the research on sleep deprivation and microbial shifts is specific enough that it can start to sound alarming if you hold it the wrong way. It isn't alarming, it's just what the body does, what the gut does, what living systems do when conditions change. Sleep deprivation has a blood signature. This is measurable, repeatable, consistent enough across studies that it functions almost as a diagnostic. Cortisol rises and stays elevated past the hours when it would normally be declining. Interleukin-6 climbs, a cytokine associated with inflammatory signaling. C-reactive protein, another inflammatory marker, increases. Natural killer cell activity drops. The immune system, which conducts a significant portion of its repair and surveillance work during sleep, finds itself running those processes against a backdrop of altered chemistry and insufficient time. The gut feels this, not abstractly, physically. The gut wall is in constant contact with the bloodstream. Immune cells patrol the mucosal lining. The cytokine environment that changes with sleep deprivation changes in the gut too, and the bacteria living against that lining are exposed to a chemically different surface than the one they occupied the night before. The mucus layer is something most people have never thought about, which makes sense because there's no reason to unless someone points at it. It lines the entire interior of the gut, a gel-like barrier produced by goblet cells in the epithelium sitting between the bacteria and the gut wall itself. It isn't static. It's produced continuously, turned over constantly. Its thickness and composition maintained by an ongoing cellular process that requires energy and signaling and time. During sleep, that maintenance runs more efficiently. The gut is not digesting actively. Blood flow to the intestinal wall is sustained. The goblet cells keep producing. The mucus layer stays robust. During sleep deprivation, the balance shifts. Inflammatory signals alter goblet cell behavior. The mucus layer thins in places, not dramatically, not all at once, but measurably. And a thinner mucus layer means that certain bacterial species, particularly those that occupy the space closest to the epithelial surface, find themselves with slightly different access to the wall than they had before. Some of them use it. The species involved vary by individual microbiome composition, which makes the research harder to reduce to a simple list. But patterns emerge across studies. Certain gram-negative bacteria, species that tend to remain at lower abundances when the gut environment is stable and the host is well rested, show increased abundance after sustained sleep deprivation. The shift isn't enormous in absolute terms, but it's consistent enough to be statistically meaningful, and it's directional. Rest suppresses these populations relative to others. Deprivation tips the balance. Firmicutes to bacteroidates ratio, a rough measure of microbial community balance that has attracted enormous research attention. Though its clinical interpretation is still contested, shifts with the sleep loss in ways that parallel what's observed in other contexts of metabolic disruption. Not identically, not through the same mechanisms, but in the same direction. What stands out to me about this is not any individual species or ratio, but the underlying logic. The microbiome is a community reading its environment. The environment changes when the host stops sleeping. The community adjusts to the changed environment. Some populations find the new conditions less hospitable, some find them more so. It isn't directed. There is no bacterial agenda. It is just populations responding to conditions the way populations always do, without knowing anything about the host's intentions or the host's schedule or the fact that the host meant to sleep but couldn't. There is a category of research finding that I find quietly compelling, which is when a biological effect reverses, when the original condition resolves. Recovery sleep, even partial recovery, even a few nights of better rest after a period of deprivation, shows measurable changes in microbial composition moving back toward the pre-deprivation baseline. Not fully in all cases, not instantly. The community takes time to restabilize, and some studies suggest that prolonged or severe sleep deprivation creates shifts that are slower to reverse than the deprivation itself was to create. But the direction changes. The conditions that favoured certain populations become less favourable again. The mucus layer recovers. Cortisol begins to find its normal rhythm. Inflammatory markers drop, and the gut quietly adjusts again. This room, wherever you are in it, has a temperature. The air has a weight. If there's a blanket over you, it has a specific pressure along the length of your body that you stopped noticing some minutes ago because the nervous system habituates to constant inputs and lets them fade. Your gut right now is in whatever configuration your recent sleep history has produced. If you've been sleeping well, the populations there are in a relatively stable state. If you haven't, something has already shifted. Not alarmingly, not irreversibly, but measurably, in a direction that rest will begin to correct. This is one of the things I find quietly remarkable about the biology of sleep. Not that deprivation causes damage, that's known, documented, not surprising, but that the body maintains such specific responsive systems in the gut that register sleep state as a chemical condition and to shift their behaviour accordingly. The microbiome is not just responding to what you eat or what pathogens you encounter. It is responding to whether you slept. And tonight you are, or you're moving toward it. The cortisol is low or lowering. Whatever inflammatory tone the day carried is beginning to settle. The goblet cells in your gut wall are doing what they do in the hours of rest, producing, maintaining, holding the barrier. The populations that expand in depletion have less reason to expand right now. Something is quieting that spent the day slightly louder than it needed to be. Not a dramatic reversal. Nothing about this is dramatic. Just the slow chemical return of conditions that suit a different configuration. The gut finding its nighttime state one population at a time in the dark without any announcement. The body reading its own rest as information. The community adjusting to what that information means. A single bacterial cell does not know how many others are nearby. It has no eyes, no spatial awareness in any sense that maps onto what awareness means for something with a nervous system. It occupies its immediate chemical environment, the few micrometers of fluid surrounding its membrane, and beyond that nothing. And yet, bacterial populations coordinate. They shift behaviour collectively, committing to actions that only make sense at scale. Biofilm formation, toxin production, bioluminescence in marine species, behaviours that a single cell initiating alone would accomplish nothing with, but that a sufficiently large population can produce effectively. The question of how they know when they are sufficiently large is, on first encounter, genuinely puzzling. The answer is chemical, and it is almost insultingly simple once you see it. Each bacterial cell continuously releases small signalling molecules into its immediate environment. These molecules, called autoinducers, though the name matters less than the mechanism, diffuse outward, diluting as they spread. In a low-density population, the concentration of autoinducers in the surrounding fluid stays low. There aren't enough cells producing them to overcome the dilution. As the population grows, more cells produce more autoinducers. The concentration rises. At some threshold, different for different species encoded in the receptor sensitivity of the bacteria themselves, the concentration becomes high enough that individual cells begin detecting it. When a cell detects that threshold, it changes gene expression. And because all cells in the population are running the same chemistry, they all cross the threshold at approximately the same moment, The population shifts together, not because anything coordinated them, because the chemistry coordinated itself. This is quorum sensing. The name comes from the political concept, a quorum being the minimum number of members required for a group to conduct official business. The analogy holds loosely. Below a certain population size, the behaviour doesn't occur. Above it, it does. What I find interesting about this, and have for a long time, is that the bacteria are not actually counting. There is no census. No individual cell knows the population size. Each cell is only measuring the local concentration of a molecule it produces, essentially asking, how much of my own signal is coming back to me? The answer to that question, it turns out, is a reliable proxy for population density. High concentration means many nearby producers. Many nearby producers means a large population. A large population means the behaviour is worth initiating. The cell is not reasoning through this chain. The receptor protein just responds to concentration, but the outcome is identical to what reasoning would produce. In the gut, quorum sensing runs continuously. Multiple species, multiple autoinducer systems, some of them species-specific and some of them readable across species. A chemical conversation about numbers and density and the conditions for collective action. The human gut is dense enough that many bacterial populations are already at or near their quorum thresholds most of the time, meaning the behaviours that quorum sensing controls, biofilm maintenance, certain metabolic commitments, gene expression shifts, are not intermittent events. They are sustained states, maintained by the continuous production of autoinducers in a space crowded enough that dilution can't keep up. Right now in the gut, molecules are accumulating, not building toward anything alarming, just accumulating the way they always do in the dark, in the warmth, in the fluid environment that bacteria have occupied for as long as the gut has existed. Each cell producing its small signal, each cell reading the concentration around it, the threshold holding, the population steady, a count being taken in the only way that has never required anyone to count. Norepinephrine is not produced in one place. This is easy to forget or to never have known, because the molecule is usually discussed in the context of the brain, the locus coeruleus, the adrenal medulla, the sympathetic nerve terminals that release it into the synapse when the body needs to sharpen attention or accelerate the heart. The neuroscience of norepinephrine is vivid and well documented and tends to dominate the picture. But norepinephrine is also synthesized by enteric neurons, the neurons embedded in the wall of the gut, part of the enteric nervous system that develops separately from the central nervous system and maintains its own local signaling infrastructure. These neurons release norepinephrine as part of normal gut function. It modulates motility. It affects the behavior of immune cells in the gut wall. It participates in the constant, mostly invisible regulation of an organ that never stops working. And it diffuses into the interstitial fluid, into the mucus layer, into the gut lumen itself, where it joins whatever other molecules are present in that dense, warm, chemically complex space. The concentration that reaches the lumen is not enormous, but it is not zero. And for a receptor protein evolved to detect norepinephrine at low concentrations, not zero is enough. The research that established this connection didn't begin with a grand hypothesis about bacterial intelligence or host microbiome communication. It began, as a lot of meaningful microbiology does, with someone noticing that bacteria behave differently under certain conditions and deciding to find out why. Vanessa Sperandio and her colleagues working in the early 2000s observed that certain enterohemorrhagic E. coli strains associated with intestinal infection, expressed virulence genes more aggressively in environments that resembled the gut than in standard laboratory culture media. The gut environment contains many things that standard media does not. One of them is norepinephrine. When they added norepinephrine to bacterial cultures, virulence gene expression increased. When they blocked the bacterial receptor responsible for detecting it, a two-component sensor system called QSEC, for quorum sensing E. coli regulator C, the effect disappeared. The bacteria stopped responding to the norepinephrine. Their virulence gene expression dropped back toward baseline. QSEC is not an adrenergic receptor in the human sense. It doesn't share structural ancestry with the alpha and beta adrenergic receptors on human cells. It arrived at norepinephrine sensitivity through an entirely independent evolutionary path. But functionally, in terms of what it does when norepinephrine is present, the outcome is similar. Detection, followed by a cellular response, followed by changed behavior. Two different architectures, the same molecule, the same consequence of its presence. What changes when QSEC activates is not one gene but many. This is where the phrase "they become different organisms" stops being rhetorical and starts being biologically precise. Gene expression is not a cosmetic feature of a cell. It is the cell's functional identity at any given moment, the set of proteins it's producing, the metabolic pathways it's running, the surface molecules it's displaying, the signals it's releasing. Change the gene expression pattern significantly enough and you have changed, in a meaningful sense, what the organism is capable of and what it is doing. When QSEC detects norepinephrine and initiates its signaling cascade, the downstream effects include changes in motility genes, flagellar expression, adhesion factors, and in pathogenic strains, the expression of proteins that allow the bacteria to attach more effectively to the gut wall. The bacterium that was swimming in the lumen can, under norepinephrine exposure, become a bacterium that anchors, that produces different surface structures, that interacts with the epithelium differently. It is the same cell, same genome, same ancestry, but it is running a different program. And the signal that switched the program came from the host. I tend to think about this in terms of what the environment actually is for a bacterium. For a human, the environment is legible, at least partially, through sensation, through perception, through the constant stream of sensory information the nervous system processes and translates into something resembling a picture of the world. The environment has textures and temperatures and distances. It has past and future in it. Because memory and anticipation are part of how it's experienced. For a bacterium, the environment is purely chemical. There is no texture, no temperature in any subjective sense, no distance. There is only the molecular composition of the immediate surroundings, what's present, at what concentration, changing in what direction. That chemical composition is the entire world, and in the gut, that chemical world includes norepinephrine, has always included it. The enteric nervous system has been producing norepinephrine in the gut for as long as vertebrates have had enteric nervous systems. Any bacterium that evolved in the gut evolved in an environment that contained this molecule. The bacteria that developed sensitivity to it, that could use it as information about host state, about whether the gut environment was shifting, had access to a signal that reliably indicated something meaningful about conditions. A rise in norepinephrine in the gut correlates with autonomic activation in the host. It correlates with stress, with disrupted sleep, with physiological strain. These are conditions under which the gut environment is also changing in other ways, immune surveillance altering, mucus dynamics shifting, motility patterns changing. A bacterium that detects norepinephrine early is detecting those changes before they've fully manifested. It has, in a sense, advance notice. The human tissue evidence for this is less extensive than the animal model evidence, partly because of the obvious difficulty of experimental access. But it exists. Norepinephrine has been detected in human intestinal tissue and luminal fluid at concentrations sufficient to activate bacterial quorum sensing systems. Studies using human gut organoids, lab-grown intestinal tissue derived from human stem cells, have shown that bacterial behavior changes in the presence of host-derived signaling molecules in ways consistent with the animal model findings. None of this is fully mapped. The research is active. The picture keeps gaining detail without yet being complete. There are mechanisms that are established and mechanisms that are proposed, and mechanisms that are suspected but not yet demonstrated. I don't want to flatten that distinction because it matters, and because one of the things that makes this area of biology genuinely interesting is precisely that it is still being worked out. What is established? Norepinephrine reaches the gut lumen. Certain bacteria have receptors that detect it. Those receptors, when activated, change gene expression. The changed gene expression produces measurably different bacterial behavior. What is still being traced? The full range of species affected. The precise concentration thresholds at which effects occur in living gut environments rather than culture conditions. The downstream consequences for host health across different contexts and timescales. Your autonomic nervous system has been active today. It is always active. That's not a dramatic statement, just a physiological one. But the particular pattern of its activity, the ratio of sympathetic to parasympathetic tone across the hours, the peaks of norepinephrine release, the moments of elevated cortisol, all of that left a chemical trace in the gut. The bacteria there read it, not all of them, not in the same way, but the chemistry was present, and what receptor proteins don't decide to respond to the molecules they were shaped by evolution to recognize. Right now, the autonomic balance is shifting. Parasympathetic tone rising, sympathetic activity quieting. The norepinephrine concentrations in the gut are following the body's lead, falling toward their nighttime level, and the bacterial populations that were reading the daytime signal are finding less of it to read. Gene expression is changing again. Not dramatically, not all at once, but the program that ran today is not the program running now. The body offered its chemistry as information, something read it, and now the body is offering something else. This quieter chemistry, this lower signal, this gradual withdrawal of the molecule that made certain behaviors possible. What that withdrawal produces in the gut is still being learned, but it is producing something, in the dark, right now, without any of it requiring you to know. Most people encounter serotonin as an explanation for something that went wrong. A doctor mentions it in the context of depression. An article describes it as the brain's happiness chemical, which is an oversimplification that has become so common it functions almost as fact now, despite being wrong in several important directions. Or someone reads about a medication that works by keeping serotonin in synapses longer and extracts from that a model of the molecule as something the brain produces and the brain uses and the brain suffers without. The brain does use serotonin. That part is accurate. Serotonin functions as a neurotransmitter in the central nervous system, involved in mood regulation, sleep architecture, appetite, cognition, social behavior, a range of functions so broad that pinning it to any single feeling or state has always been an oversimplification. But roughly 90% of the body's serotonin is not in the brain. It is in the gut, produced there, stored there, acting there on tissue that has its own serotonin receptors, its own serotonin transporter proteins, its own relationship with this molecule that predates its role in anything resembling mood by an enormous stretch of evolutionary time. The cells that produce most of the gut's serotonin are called enterochromaffin cells. They're embedded in the epithelial lining of the intestine, not neurons, not immune cells, but epithelial cells that have taken on a specialized secretory function, sitting at the interface between the gut lumen and the tissue beneath it, reading the chemical environment on one side and releasing serotonin into the tissue on the other. What they're reading partly is the microbiome, short-chain fatty acids produced when gut bacteria ferment dietary fiber stimulate enterochromaffin cells to produce serotonin. This has been demonstrated in animal models with enough consistency that it's considered an established pathway rather than a speculative one. Germ-free mice, raised without any gut microbiome, have significantly lower levels of circulating serotonin than mice with normal microbial colonization. When those germ-free mice are colonized with specific bacterial strains, particularly spore-forming bacteria from the Clostridia class, serotonin levels rise toward normal ranges. The bacteria are not producing serotonin. They are producing the conditions under which enterochromaffin cells produce more of it. The fermentation byproducts are the signal. The enterochromaffin cells are reading that signal and responding, and what they release when they respond touches everything downstream. Serotonin in the gut does not primarily regulate mood. That function belongs to the small fraction of the body's serotonin that crosses into central nervous system tissue. And even there, the relationship between serotonin levels and mood states is far more complex and contested than popular accounts suggest. In the gut, serotonin regulates movement. Gut motility, the coordinated muscular contractions that move contents through the intestine, is substantially controlled by serotonin acting on enteric neurons. When enterochromaffin cells release serotonin in response to luminal stimulation, it activates serotonin receptors on the nerve cells in the gut wall, triggering the peristaltic reflex that pushes things along. Too little serotonin signaling in the gut and motility slows. Too much and it accelerates. This is why medications that affect serotonin signaling, even those developed primarily for psychiatric purposes, often have pronounced gastrointestinal effects. The gut has more serotonin receptors than the brain. It is, in a pharmacological sense, more serotonergic than the organ serotonin is usually associated with. Now, the bacteria. Some gut bacterial species possess enzymatic machinery that acts on tryptophan, the amino acid that serves as the precursor from which serotonin is synthesized. Human cells convert tryptophan to serotonin through a two-step enzymatic process. Bacteria can intercept tryptophan before that conversion happens, metabolizing it through their own pathways into different compounds, some of which have their own biological activity, some of which are essentially removed from the serotonin synthesis route. The implications of this are still being worked out in detail, but the basic logic is clear. If bacteria are consuming tryptophan in the gut, the availability of that precursor for serotonin synthesis changes. The enterochromaffin cells are drawing from the same tryptophan pool that the bacteria are drawing from. The bacterial population's size, composition, and metabolic activity directly influences how much raw material is available for serotonin production. This is not the bacteria regulating serotonin. There is no bacterial intention here, no coordination, no awareness of what serotonin is or does. It is the bacteria running their own metabolism in an environment that happens to be shared with a host that depends on tryptophan for a signaling molecule the bacteria know nothing about. The competition for a resource, without either party knowing there is a competition. There is also evidence, less established, still actively studied, that some bacterial species possess receptors that respond to serotonin itself, that they detect serotonin concentrations in the gut lumen and adjust their behavior accordingly. Motility, biofilm formation, gene expression patterns, shifting in response to a molecule the bacteria did not produce and cannot use in the way the host uses it, but can apparently detect and respond to. What stands out most to me about this, when I sit with it, is the layering. Bacteria influence how much serotonin the gut produces. The serotonin then affects the gut environment, motility, immune tone, inflammation levels, and that changed gut environment feeds back to influence bacterial populations. The bacteria that altered the serotonin landscape now live in a gut shaped by the serotonin landscape they helped create. The signal goes around, not in a neat circle, in something messier and more continuous than that. A loop that never closes cleanly, that involves constant adjustment from both directions, that was running before anyone thought to describe it as a loop at all. There is a particular quality to the hour before sleep that most people recognize without having words for it. Not drowsiness exactly, that comes later. Something earlier than that, a slight softening of the edges of things, a reduction in the urgency that thoughts carry during waking hours, as though the mind is beginning to release its grip on whatever it was holding. The body is doing things during this transition that involves serotonin. The synthesis of melatonin from serotonin in the pineal gland, the shift in neurotransmitter balance that sleep onset requires, the quiet withdrawal from the waking serotonergic tone that alertness depends on. The gut is doing things too. Motility slowing, immune surveillance shifting into a different pattern. The enterochromaffin cells reading a different luminal environment than the one they occupied an hour ago, because the microbiome's metabolic activity has its own circadian rhythm, its own nighttime configuration, producing different concentrations of the short-chain fatty acids that influence serotonin production. The serotonin the gut is managing right now is not quite the same as the serotonin it was managing this afternoon. The bacterial community producing the conditions for its synthesis is not quite the same community. The host state, which the bacteria have been reading all day through norepinephrine and cortisol, and the other chemical signals that leak into the lumen, has changed. And the microbiome is responding to that change in its own way, which feeds back into the serotonin the gut produces, which affects how the gut wall behaves, which shapes the environment the bacteria continue to inhabit. The molecule was not built with a second audience in mind. There was no design, there was just chemistry, present in a space that other chemistry also occupied, for long enough that relationships formed without anyone choosing them. The bacteria in your gut right now are not thinking about your serotonin. They are not thinking about anything. They are metabolizing tryptophan because tryptophan is there and their enzymes recognize it. They are responding to short-chain fatty acid gradients because their receptors detect those gradients. They are doing what their biology has always done in this environment, in the dark, in the warmth, in the slow chemistry of a gut winding down for the night. And somewhere in that activity, in the space between what they produce and what your enterochromaffin cells read from it, your gut is deciding how much serotonin to make. The signal moves. The gut responds. Something in you shifts slightly toward whatever sleep requires. The second audience never knew it was listening, and it doesn't need to. 1 cell thick. In places, that's all there is between the interior of the gut, with its 38 trillion microbial residents, its fermenting contents, its dense and constant chemical activity, and the bloodstream. 1 cell. A single epithelial cell, maybe 20 micrometers across, its top surface facing the lumen, and its bottom surface resting against the tissue that leads, eventually, to capillaries, to circulation to the rest of the body. 20 micrometers is smaller than you can see, smaller than the period at the end of a sentence if you printed this and held it at arm's length. The barrier between the microbiome and the blood is that thin, and yet it works, most of the time, which is when you stop to consider what it's doing, quietly extraordinary. The epithelial layer is not passive. It is not a wall that molecules either breach or don't. It is an active surface covered in transport proteins, receptor proteins, junction proteins that control the spaces between cells, immune signaling molecules that communicate downward into the tissue below. The epithelial cells are reading what arrives at their luminal surface, making decisions, in the only sense that cells make decisions, which is chemical and without awareness, about what crosses and what doesn't, what gets modified in transit, and what passes through the unchanged. Some things the epithelial layer allows. Some things it chemically alters as they cross. Some things it blocks. And some things it produces itself in response to what the microbiome presents at its surface, sending signals downward into the tissue that the microbiome never generated, and the bloodstream receives as though they came from nowhere. This is what makes the epithelial layer an interpreter rather than a wall. An interpreter doesn't just transmit. It receives something in one language and produces something in another. What comes out of an interpretation is related to what went in, but it's not identical. Something is always added, something always lost in the act of translation. The microbiome produces short-chain fatty acids, butyrate, propionate, acetate, molecules that gut bacteria generate as fermentation byproducts, released into the lumen in concentrations that reflect the composition and metabolic state of the community. These molecules reach the epithelial surface, and what happens there is not simple passage. Butyrate, for example, is actively taken up by colonocytes, the epithelial cells of the colon, and used as their primary energy source. The colonocytes metabolize most of it before it can reach the bloodstream. What enters the portal blood is a fraction of what the bacteria produced, chemically transformed by cells that used it to power their own function. The bacterium produced butyrate as a byproduct of eating fibre. The epithelial cell consumed that butyrate to survive, and the immune cells below the epithelium, which butyrate also modulates when it reaches them, received whatever was left. 3 different biological relationships with the same molecule, occurring in sequence, each one changing what the next one encounters. The tight junctions are worth a moment. Between epithelial cells there are protein complexes, claudins, occludins, zona occludens proteins, that seal the spaces between adjacent cells. These junctions are dynamic. They tighten and loosen in response to signals from the gut environment, from the immune system, from the microbiome itself. Certain bacterial metabolites strengthen tight junction integrity. Others, under different conditions, reduce it. A looser tight junction means more can pass between cells rather than through them, the paracellular route, bypassing the transport proteins and the metabolic processing that transcellular passage involves. Things cross that wouldn't have crossed otherwise. In concentrations that wouldn't otherwise occur, the microbiome influences the tightness of the junctions. The tightness of the junctions influences what the microbiome can send across. The epithelial layer is not a fixed threshold, but a variable one, and part of what sets the variable is the population living on the other side of it. I find something genuinely hard to resolve about this. Not distressing, just open. The idea that the boundary between the microbiome and the body proper is negotiated rather than fixed. That what the blood receives from the gut is not simply what the gut contains, but a version of it, translated, filtered, some modified, sometimes amplified, and sometimes reduced by a layer of cells that is itself being shaped by the microbiome it's interpreting. What the body hears is not exactly what the microbiome said, but it's not entirely different either. Right now, the epithelial cells in your gut are doing this, quietly, without any sensation reaching you from it. The tight junctions are at whatever tension the current gut environment produces. The transport proteins are running their gradients. Butyrate is being consumed by colonocytes in the dark of your colon, powering cells you will never feel, maintaining a surface you will never directly sense. The microbiome is presenting itself at the border. The border is interpreting what it receives, and somewhere below that, in the tissue, in the capillaries, in the circulation that touches every organ, the body is receiving a translation, incomplete, modified, filtered through a layer one cell thick in places. Hearing something, not exactly what was said, but enough. Nobody planned this. That feels like the right place to begin, before the biology gets specific, before the receptor names arrive and the metabolic pathways unfold. Before any of it, nobody planned this. The bacteria were not evolving toward usefulness. The human receptors were not waiting for bacterial molecules. There was no collaboration, no co-design, no mutual recognition of shared interest. There was just proximity, time, chemistry that happened to fit. Short-chain fatty acids are small, 2 to 6 carbon atoms in the chain, an acidic group at one end, acetate, propionate, butyrate, the 3 most studied, the 3 most abundant in the human gut. They are produced when anaerobic bacteria ferment dietary fibre in the colon, not as signals, not as gifts, as waste essentially. The bacterial equivalent of exhaled breath, a byproduct of eating released into the environment because it has no further use to the cell that produced it. The molecules diffuse into the gut lumen, toward the epithelial surface, into the mucus layer. Some get taken up by colonocytes immediately metabolized as energy. Some pass through the epithelial layer into circulation. Some remain in the lumen, part of the dense chemical mixture that the gut environment always is. And some reach G-protein coupled receptors on human cells. G-protein coupled receptors are among the most ancient and abundant receptor proteins in animal biology. There are over 800 distinct ones encoded in the human genome. They span the cell membrane, with one end facing outward and one end facing into the cell's interior, and they operate through a shared mechanism. A molecule binds on the outside, the receptor changes shape, and that shape change activates a G protein on the inside of Muo, which then initiates a signaling cascade that changes what the cell does. Vision uses them, smell uses them, hormone signaling uses them. The heart's response to adrenaline uses them. They are, in a structural sense, one of biology's most successful solutions to the problem of detecting something outside and responding to it inside. Two of them, GPR41 and GPR43, now often called free fatty acid receptors, FFAR3 and FFAR2, respond to short-chain fatty acids. They are found on gut epithelial cells, on immune cells, on enteroendocrine cells that release hormones affecting appetite and metabolism, on fat cells, on cells lining blood vessels. When short-chain fatty acids bind to them, the cells they sit on change behavior. Immune cells alter their inflammatory state. Enteroendocrine cells release peptides that signal satiety to the brain. Fat cells modulate their activity. Gut motility shifts. Here's the part that stays with me. FFAR2 and FFAR3 did not evolve to detect bacterial fermentation byproducts. Or, more precisely, we cannot say that it evolved for that purpose because evolution doesn't work in terms of purpose. What we can say is that these receptors are shaped to respond to short-chain fatty acids, and that in the gut, the primary source of short-chain fatty acids is bacterial fermentation. Whether the receptors develop their sensitivity in response to that microbial presence over evolutionary time, or whether the sensitivity existed first and the bacteria simply happened to produce something that fit, that question doesn't have a clean answer yet. What's clear is that the fit exists. The bacterial byproduct and the human receptor are compatible in a way that produces consistent, measurable biological effects. The bacteria exhale something. The human cells inhale it. Not intentionally, not reciprocally, just because the molecule was present and the receptor was shaped to notice it. Somewhere in your colon right now, fermentation is happening. Probably slowly given the hour. The microbiome has its own circadian tempo, and its fermentative activity is not constant across the day. But it doesn't stop. Anaerobic bacteria don't require waking hours. They require fiber and the right conditions, both of which the colon provides continuously. Acetate is being released. Propionate, butyrate, small molecules drifting towards surfaces that will receive them without knowing they're coming. The receptors are there, holding their shape, ready to respond to a geometry that bacteria never evolved to produce and human cells never evolved to request. The fluency was borrowed without asking. It works anyway. There is a way to know what the microbiome does that doesn't involve studying it directly. You remove it entirely. You raise an animal from birth in a sterile environment, filtered air, irradiated food, no contact with any surface that carries living microorganisms, and you watch what develops in the absence of everything the gut would normally contain. What grows differently, what fails to develop, what the body builds when the conversation it was expecting never begins. Germ-free animal models have existed since the mid-20th century. Mice, rats, zebrafish, pigs, various species raised in isolators, their guts colonized by nothing, their immune systems encountering no microbial signals from birth. The research that has come from these models is, in aggregate, one of the more quietly unsettling bodies of evidence in modern biology. Not because the findings are dramatic, because they're specific. The body, it turned out, was expecting something, and the places where development goes differently in its absence are not random. The gut wall is thinner in germ-free mice. The villi, the finger-like projections that extend from the intestinal surface and vastly increase its absorptive area, are shorter and less dense. The crypts, the recessed regions between villi where stem cells divide and produce new epithelial cells, show altered proliferation rates. The mucus layer is different in composition and thickness. The tight junctions between epithelial cells behave differently. These are not subtle findings, they are structural. Changes in the physical architecture of the gut that are visible under a microscope and measurable across multiple studies, multiple species, multiple laboratory conditions. the gut wall that develops without microbial signals is not a normal gut wall that simply lacks bacteria. It is a different gut wall, one that formed in the absence of signals it needed to form correctly. This matters because it tells you something about what the body's developmental program assumed. The instructions encoded in the genome for building a gut wall were written in an evolutionary context where bacteria were always present. The program has steps that depend on microbial signals to execute properly, not as a vulnerability, not as a flaw in the design, but because for the entire evolutionary history in which that program was being refined, the signals were always there. There was never a selection pressure to build a gut wall without them. The immune system findings are, if anything, more striking. Germ-free mice have fewer immune cells in the gut-associated lymphoid tissue, the Peyer's patches, the mesenteric lymph nodes, the diffuse collections of immune cells that line the intestinal wall. The cells they do have are less mature, less functionally calibrated. The balance between pro-inflammatory and anti-inflammatory immune responses is skewed. Regulatory T cells, which are essential for preventing immune overreaction and maintaining tolerance to harmless antigens, including food proteins and the body's own tissues, are reduced in number and in function. When germ-free mice are colonized with normal gut bacteria, regulatory T cell populations begin to recover. The immune architecture moves towards something closer to normal, but the recovery is not complete, and it is not uniform across developmental stages. Mice colonized as adults recover differently from mice colonized in early life. There are windows, specific periods during immune development when microbial signals are required to calibrate the system, and if those windows close without the signals arriving, the calibration that didn't happen does not simply occur later when the signals do arrive. The immune system learned something during those windows, or failed to, and what it failed to learn during the window it does not go back and learn afterward. The stress hormone data is, to me, the part that sits most uneasily. Germ-free mice show dysregulated HPA axis responses, the hypothalamic pituitary adrenal axis, which governs cortisol and corticosterone release in response to stress. When exposed to a stressor, germ-free animals mount a larger hormonal response than conventionally colonized animals. Their stress response is not just different, it is, by conventional measures, more reactive, less regulated, less able to return to baseline efficiently once the stressor has passed. When germ-free mice are colonized with gut bacteria shortly after birth, the HPA axis dysregulation partially corrects. But again, and this is the part that holds my attention, only if the colonization happens early enough. There is a postnatal window during which microbial colonization shapes HPA axis calibration. Outside that window, colonization with normal gut bacteria does not normalize the stress response. The window closed. The calibration that was meant to happen during it did not happen. The axis runs differently and continues to run differently for the animal's life. What this implies, carefully and without overclaiming, is that the body's stress regulatory system developed in a context where gut bacteria were present from birth, and that its calibration during early life depends in some measurable way on signals from those bacteria. A gut that goes quiet at the wrong time doesn't just produce a different gut. It may produce a different nervous system. There is something I find worth sitting with here, and it's not anxiety. It's more like a recalibration of what the self is. The standard model of the individual organism treats the body as the unit of biological identity. Skin defines the boundary. What's inside is you, what's outside isn't. The genome is yours, the microbes are passengers. But the germ-free data suggests that the body's developmental program cannot run correctly without the passengers, that the skin boundary doesn't cleanly map onto the biological process of becoming. The immune system, the gut architecture, the stress regulatory axis, all of these developed in an environment that included microbial signals, and all of them develop differently when those signals are absent. The body was not designed to develop alone. The design assumed company. Right now you've been colonized since birth, or close to it. The developmental windows have opened and closed with bacteria present. Whatever their presence shaped in your gut wall, your immune calibration, your early stress axis development, that shaping happened. It is part of what you are now, built into the architecture of body that was never, in its entire developmental history, without them. There is something quietly grounding in this if you hold it the right way. Not that the microbiome made you, but that the body never developed in a way that required you to be separate from it. The conversation started very early, before you had language for anything, before you had language at all. And the body that formed from that conversation, the gut wall with its bacterial-dependent architecture, the immune system calibrated against microbial signals, the nervous system that developed in an environment your microbiome helped create. That is the body breathing now, in the dark, in this room, slowing toward sleep. It was built with them in it. It is running still the same way. The immune system is, among other things, a reading system. Not reading in the way that implies comprehension. Nothing in the immune system understands what it encounters, but it reads in the older sense, in the sense of scanning surfaces for patterns, comparing what's present against what's known, and responding differently depending on whether the pattern matches or doesn't. Every cell in the body carries surface molecules that mark it as belonging. Human leukocyte antigens, proteins that display fragments of what's happening inside the cell, allowing immune surveillance to check whether the interior chemistry is normal. Self-associated molecular patterns that immune cells are trained during development to leave alone. The immune system learns what the body looks like, and then it looks constantly for things that don't look like that. Bacteria have surfaces too, lipopolysaccharides on gram-negative bacteria, peptidoglycan in the cell walls of gram-positive species, flagellin proteins in the whipping tails some bacteria use to move. These are structures the immune system has pattern recognition receptors specifically designed to detect. Toll-like receptors, NOD-like receptors, a family of proteins whose entire function is to recognize molecular signatures associated with microbial life and trigger an immune response when they're found. This system works. It is why the immune system responds to pathogens, why infection triggers inflammation, why the body mounts a defense rather than simply accommodating whatever arrives. But the gut contains trillions of bacteria. If the immune system responded to every microbial surface molecule with the full machinery of defense, the gut would be in a state of permanent inflammation. The epithelial layer would be constantly under assault from the immune cells meant to protect it. The body would be, in a measurable sense, attacking its own interior continuously. It doesn't do this, and the reason it doesn't is not simply that the immune system ignores gut bacteria. It recognizes them, and then, for many of them, it stands down. The standing down is learned. During early life, during those same developmental windows that appear elsewhere in this series, the periods when the body is calibrating systems against the microbial environment it finds itself in, immune cells in the gut encounter commensal bacteria, and learn, through a process involving regulatory T cells and telerogenic dendritic cells and a specific cytokine environment, to distinguish them from pathogens. The surface molecules are read, the patterns are logged. The response is modulated downward, not to zero, but to a level of quiet surveillance rather than active attack. This tolerance is not permanent in the sense of being fixed. It requires maintenance. The microbial signals that established it need to continue arriving for it to hold. Germ-free animals, as the previous chapter touched on, develop immune systems that are improperly calibrated, partly because this telerogenic learning never happened. The immune system was never trained on the full range of commensal surfaces. It doesn't know what to ignore. Now the strange part. Some bacteria have surface molecules that don't merely avoid triggering immune responses. They actively resemble host molecules closely enough that immune cells read them as self rather than as microbial. The polysaccharide capsules of certain bacteroid species, for example, contain molecular structures that mimic mammalian carbohydrate patterns closely enough to influence how immune cells categorize them. Not perfectly, not identically, but closely enough that the pattern recognition machinery running its comparison lands on something close to recognition rather than something close to alarm, the immune cell encounters the surface, reads it, finds something that matches, approximately, the molecular language of the host, and responds as though what it found belongs here. I want to be careful about the word mimicry. Mimicry implies intent. It implies one party deliberately copying another for advantage. And in evolutionary biology, the word is often used for systems where that functional outcome is so consistent and specific that it strains to describe it any other way. But the bacteria are not copying. There is no bacterial awareness of what human surface molecules look like or what resembling them achieves. What happened over enormous stretches of time is that bacteria living in an environment shaped by mammalian immune systems were under selection pressure to avoid triggering those systems, and the ones whose surface chemistry happened to resemble host molecules closely enough to reduce immune activation survived better and reproduced more. The surface chemistry that remains is the chemistry that worked, not designed to work, just the residue of what wasn't destroyed. And on the other side of the same process, the immune system developed its recognition machinery in an environment that always contained these bacteria, the pattern recognition receptors, the telerogenic pathways, the self versus non-self distinction. All of it developed while these bacteria were present, with these surface molecules looking the way they look. Two systems, the same evolutionary pressures, the same chemical environment, arriving independently at a shared molecular language neither of them invented. Right now, immune cells are patrolling the lining of your gut, scanning surfaces, reading patterns, encountering bacterial molecules that have been present in the gut environment for longer than the human immune system has existed in its current form. Some of those encounters produce signals of danger. Most don't. Most produce something quieter, a kind of recognition without alarm, a reading that finds nothing worth responding to and moves on. Not trust exactly, the immune system doesn't trust, but something functionally close to it, built from the same material as everything else in this series, proximity, time, and the slow directionless pressure of surviving in the same space as something you will never fully understand. Start with the numbers, because the numbers matter here in a way that isn't about drama. The human genome contains approximately 20,000 protein-coding genes. This is fewer than a grape, fewer than a water flea. The number surprised biologists when the Human Genome Project produced its first draft count in the early 2000s. The expectation had been something closer to 100,000, given the complexity of what those genes were assumed to need to produce. 20,000 turned out to be enough, partly because of the sophistication of gene regulation, the ways a limited set of genes can produce enormous functional variety depending on when and where and how much they're expressed. 20,000 is a real number. It represents something, and somewhere between 2 million and 20 million is the number of protein-coding genes in the collective genome of the human microbiome, what researchers call the metagenome, the aggregate genetic content of all the microbial species that inhabit a human body. The range is wide because the microbiome varies between individuals, because sequencing technology continues to improve and find genes previously missed, because the number genuinely isn't fixed. But even the lower bound is 100 times larger than the human genome. The body you think of as yours carries a genetic library roughly 100 times the size of the one encoded in your cells. Most of those microbial genes do things human genes cannot. This isn't a gap in human biology that evolution failed to fill. It is, in a very specific sense, how human biology was shaped. In an environment where those microbial functions were always present, always available, the human genome never needed to develop them independently. The functions were covered by residents whose presence was, for the entire evolutionary history of the human lineage, as reliable as the presence of oxygen or water, or the molecular building blocks of food. Bile acid metabolism is one clear example. Primary bile acids are produced by the liver and released into the small intestine to help digest fats. In the colon, gut bacteria convert those primary bile acids into secondary bile acids, compounds with different properties, different receptor affinities, different effects on gut motility and immune function and glucose metabolism. Human cells do not perform this conversion. The enzymes required are encoded in microbial genomes, present in the gut in sufficient abundance that the conversion happens reliably, continuously, as a normal part of digestive physiology. The body expects this to happen. Its metabolic pathways downstream of bile acid processing assume that secondary bile acids will be present. If the relevant bacterial populations are disrupted by antibiotics, by significant dietary change, by illness, the secondary bile acid profile of the gut shifts, and the downstream physiology shifts with it, in ways that are measurable and sometimes clinically significant. The body was not designed to produce these compounds itself. It was designed in a context where something else produced them, reliably, without being asked. Vitamin K2 follows a similar logic. Certain B vitamins, amino acid conversions that the gut relies on for downstream synthesis. The human body performs these functions, when it performs them at all, with microbial assistance so old and so consistent that the assistance is simply assumed in the biology. The metabolic map of a human body is, in places, a map drawn with microbial contributions included. Not labelled as contributions, just present, the way a foundation is present in a building without being visible in the rooms above it. There is something I find difficult to name cleanly about this. It isn't symbiosis in the greeting card sense, mutual benefit, cooperation, two organisms helping each other. The bacteria are not helping. They are running their own metabolic programs in the environment they occupy, and those programs happen to produce outputs the human body has learned over evolutionary time to depend on. The dependency formed because it could form, because the outputs were reliably there, because evolution moves toward available resources without planning to. But the result of that unplanned movement is a body that cannot fully function on its own genome, that relies, for specific metabolic steps, on genes it doesn't carry. The metagenome concept emerged in the early 2000s, growing from advances in sequencing technology that allowed researchers to sequence genetic material directly from environmental samples, from soil, from ocean water, from the gut, without culturing individual species first. Before metagenomic sequencing, gut microbiome research was limited to the species that could be grown in a laboratory, which excluded the majority of gut bacteria, most of which are strict anaerobes that die on contact with oxygen. What emerged from that picture was not a catalogue of foreign material sitting alongside the human body. It was something harder to categorize, a genetic landscape in which human genes and microbial genes together constitute the functional whole. Neither set sufficient alone, both sets operating in the same biological space, producing outputs that feed into each other so thoroughly that the boundary between host metabolism and microbial metabolism blurs in ways that resist clean description. I tend to think this is where the concept of biological selfhood gets genuinely interesting rather than genuinely troubling, though I understand why some people land on the troubling side first. The self that biology has always assumed, the individual organism bounded by skin, defined by its genome, is real. You are a real biological entity. The genome in your cells is yours in a meaningful sense. But that entity developed in an environment that always included a microbial community, and the metabolic functions of that community are woven into your physiology at a level that makes clean separation not just difficult, but, in certain respects, incoherent. You're not your microbiome, but you have never been entirely separable from it either. Right now in your gut, microbial genes are being expressed. Enzymes encoded in bacterial genomes are running reactions that your own genome has no instructions for. Compounds are being produced that your cells will receive and use and require, without any marker indicating where they came from, without any cellular mechanism to distinguish a metabolite produced by a human cell from one produced by a bacterium. The body doesn't make that distinction because it never needed to. The distinction arrived later, with categories, with the concept of self and other, host and inhabitant, mine and not mine. The biology was already running before the categories existed to describe it. It is still running now, in the dark, without the categories. in the warm, quiet interior of a body that was never, in any biological moment it has ever occupied, entirely alone. The room is darker now than it was an hour ago, not because anything changed in the room, because something changed in you, the pupil dilating, the retinal sensitivity shifting, the visual cortex adjusting its interpretation of low-light input. The darkness feels deeper partly because the body is moving toward a state in which darkness is the appropriate condition. The nervous system is not fighting the dark. It is, in some cellular sense, expecting it. Melatonin has been rising since the light began to dim. It will keep rising through the first hours of sleep, peaking somewhere in the middle of the night and then beginning its long decline toward morning. This is the most familiar piece of the body's circadian biology, the hormone most people have heard of, the one associated most directly with sleep onset, with the feeling of being drawn downward into rest. But melatonin is not where the circadian story begins. It is one output of a system that runs at every level of the body simultaneously, in organs, in immune cells, in the epithelial lining of the gut, and in the microbiome. The discovery that gut bacteria have circadian rhythms was not, in retrospect, surprising. But it was not obvious either, because bacteria don't have the molecular clock machinery that animal cells use to track time. Animal circadian clocks run on interlocking feedback loops of specific proteins, clock, BMAL1, PER, CRY, that cycle with a period of approximately 24 hours and drive rhythmic gene expression in nearly every cell type in the body. Bacteria don't have these proteins. There was no reason to expect them to show 24-hour rhythms. And yet, when researchers began taking time series measurements of gut microbiome composition, sampling the gut contents of mice at multiple points across the day and night, they found that different bacterial populations rose and fell in abundance with striking regularity across the 24-hour cycle. Firmicute species showed peak abundance at certain hours, bacteroidate species at others. The overall composition of the community at any given hour was measurably different from its composition 6 hours earlier or 6 hours later. The bacteria were not running their own clocks. They were being entrained by the host. Feeding schedules, light exposure, the rhythmic output of host hormones and gut motility patterns, all of these created a gut environment that changed predictably across the day. And the bacterial populations were shifting in response to that changing environment, their abundance patterns tracking the host's rhythms without possessing any intrinsic timekeeping mechanism of their own. The host set the tempo. The microbiome followed it. But the relationship is not simply one of entrainment flowing in one direction. The metabolic outputs of the microbiome, the short-chain fatty acids, the tryptophan metabolites, the secondary bile acids, the immune-modulating compounds, are produced in quantities that vary across the day according to the bacterial population rhythms, which means the body receives different concentrations of these compounds at different hours, and the body's own circadian machinery in the gut wall and in the liver and in metabolic tissues throughout has incorporated these rhythmic microbial signals into its own timing. Germ-free mice show disrupted circadian gene expression in the gut and liver, not just in the gut wall, but in metabolic tissues remote from the gut, tissues that receive microbial signals through circulation. Their peripheral clocks run differently without the rhythmic chemical input that the microbiome normally provides. When the microbiome is restored, circadian gene expression partially normalizes. The host is not just setting the microbiome's rhythm. The microbiome is, in part, sustaining the host's rhythm. Two systems, circadian co-regulation flowing in both directions, each partly dependent on the other's consistency to maintain its own. What the microbiome does differently at night is worth staying with. Fermentative activity continues. Bacteria don't stop metabolizing because the host is sleeping, but the substrate availability changes. Gut motility slows during sleep, which means transit time lengthens, which means the time bacterial populations have to interact with luminal contents increases. Fermentation of fiber that moved quickly through the gut during the day now happens more slowly, in a different luminal environment, with different bacterial populations dominant. Butyrate production, in some studies, shows a nocturnal peak. This matters because butyrate is the primary energy source for colonocytes, the epithelial cells of the colon, and colonocyte proliferation, the renewal of the gut lining, shows its own circadian pattern, with activity concentrated during rest periods. The microbiome's nocturnal butyrate production aligns with the gut wall's nocturnal repair cycle. Whether this alignment was selected for, or whether it is an emergent property of two systems that developed in proximity for long enough to fall into synchrony, that question doesn't have a clean answer yet. But the alignment is there. The gut repairs itself at night, partly because the microbiome is producing at night the fuel that repair requires. Tryptophan metabolism shifts too. The bacterial handling of tryptophan, and therefore the availability of tryptophan for serotonin synthesis, and therefore the availability of serotonin for melatonin synthesis, follows rhythmic patterns that vary with microbial community composition across the day. In the hours before and during sleep, certain bacterial metabolic pathways that compete with the serotonin synthesis route are less active. The tryptophan that might be shunted into other microbial metabolic channels during waking hours is, in the nocturnal gut environment, somewhat more available for the host's own chemistry. This is not the microbiome generously providing melatonin precursors. There is no provision. There is just the rhythmic activity of a community whose metabolic tempo happens to create, in the hours the host needs it most, conditions slightly more favourable for the chemistry of sleep. The melatonin rising in your bloodstream right now exists at the intersection of your pineal gland's response to darkness and a tryptophan availability that the gut microbiome has been quietly shaping all evening. I want to end here, in this room, in this body, in this specific biological moment, because that is where the series began and where it belongs. Somewhere in your gut, right now, the populations are in their nocturnal configuration. The species that peak during waking hours are at lower abundance. The ones that do their particular work during rest are running their chemistry in the warm dark of your colon, producing compounds that your gut wall will absorb and your circulation will carry and your biology will use in ways neither the bacteria nor the body chose or designed. The butyrate is being produced. The colonocytes are proliferating. The tight junctions are at their nighttime tension. The immune cells in the gut-associated lymphoid tissue are doing the surveillance work that sleep allows more time for, running their quiet inspection of the surfaces the microbiome presents to them, reading patterns, standing down from what they recognize, maintaining the tolerance that took years to learn. Everything in this series has been happening tonight, not as a consequence of listening to it. It was happening before you pressed play. It was happening before you knew any of the words for it. It has been happening every night of your life, in configurations shaped by what you ate, how much you slept, and what stress you carried, and what the bacteria encountered when they arrived in your gut in the first days after you were born. The microbiome does not know you are sleeping. It doesn't know anything, in the sense that knowing requires, but its chemistry is responding to the chemistry of a host in its rest state. Lower cortisol, lower norepinephrine in the gut lumen, shifted motility, longer transit times, the particular immune tone of a body that has lowered its daytime vigilance and turned toward repair is responding the way it has always responded to these conditions, across every night of your life, across the evolutionary history of every human gut that came before yours. And slowly, across the hours ahead, it will begin to shift again, not abruptly, not at any specific moment. But somewhere in the deep hours of night, the populations will begin their slow movement toward morning configuration. The species that peak during waking hours beginning their quiet climb, the metabolic tempo changing, the chemical outputs tilting toward what a waking body will need. It is preparing something, not for you exactly, not with you in mind, just running its rhythms, the way it always has, in the dark, in the warmth, in the body that carries it, and that it, in its quiet way, helps carry in return. The light is off. Something is still working. It always was.